Welcome back, everyone. Thank you so much for joining us today for our first talks since the summer break. And we are very, very happy to have two wonderful speakers, sorry, speakers with us today. The AI for Women's Health group in the Alan Turing Institute is coordinated by myself, Emma, Annalisa, and Kristen. And Annalisa and Kristen, well, I will introduce the first speaker, Eva, and then Kristen will introduce our second speaker, Roland, and Annalisa will chair the questions at the end. So thank you very much, everyone, for joining, and thank you for our speakers to be here. So I'll start by introducing Eva. So Eva holds a bachelor's and master's degree in computer science from the University of Ljubljana. Her master's thesis focused on the segmentation and alignment of uterine shapes in 3D ultrasound data. And currently, Eva is pursuing her PhD at the University of Ljubljana, where she continues to expand her research in the field of computer vision and visualization. So Eva is talking to us today about a very interesting topic and paper that also includes, and this is quite important to note, an open source data set, which is quite rare in this field. Eva, the floor is yours.
Thank you. So I will try to share the presentation and hopefully third time it works. Just let me know. Can you see it normally? Okay, perfect. Okay, so hello. My name is Eva, as Bianca already said, and I'm a PhD student at the Faculty of Computer and Information Science here at the University of Ljubljana. Today I'm going to present you the work that I did for my master's thesis, and it's a project that we're still working on. Now, it was an interdisciplinary project, and we worked closely with a gynecologist who actually came to us with his problem, and we were very happy that we could give him a solution, at least a part of the solution. So my plan for today will be to first give you some background on the clinical problem that we're addressing and what the goals of our work were. And then I'll go over our methods, but I'll keep the technical details brief, and I'll focus more on the overall approach and the idea. But if anybody has any more in-depth questions, I'll be happy to answer them. So the project is called the Automatic Segmentation and Alignment of Uterine Shapes from 3D Ultrasound Data. And it is a part of the Normal Uterus Assessment Study, or NURSE, as we call it. And this is a large collaborative European project that's involving 15 expert gynecology centers. And their goal is to use the real-world data and create first truly evidence-based definition of a normal uterus. And our work somehow provides the automated tools to make that analysis possible on a larger scale. So let's start with the core problem, which is what is actually a normal uterus. So in medicine, the normal values for any parameter or any organ, for example, are typically based on measurements that we get from a large, healthy population. But for uterine morphology, a precise technical definition of a normal uterus is still lacking, or better, it does not yet exist. Some classification systems have been developed, but they are interestingly inconsistent with each other. So they use different measurement techniques and descriptive criteria, which means that a uterus that is considered normal under one system might be classified differently under another, which can lead to diagnostic confusion and potential overtreatment or undertreatment of certain conditions. But the most critical issue is that these classification criteria were set almost arbitrarily, just by a committee consensus, and were not really based on real-world data from a healthy population. And this means that the very foundation of uterine classification is not really built on evidence. So to address this gap, the NURSE study was initiated. So again, as I already mentioned, this is a large multi-center prospective cohort study that has one goal, which is to create the first evidence-based definition of a normal uterus. And the study design was very rigorous to ensure this high quality and reliable data. And they recruited 442 nulliparous women, meaning women aged 18 to 35, that had regular cycles and have never given birth, so that we can... Sorry, all good? So that we can establish a healthy baseline that is free from any changes that are related to pregnancy. And to ensure consistency across all 15 centers, every ultrasound was performed during a very specific window of the proliferative phase, when the uterine lining is clearest and the measurements are the most accurate. Now, from these acquired scans, the clinical team had to manually measure dozens of specific lengths, thicknesses, and angles. And the result of this work is a detailed statistical model. So for the first time, we have a set of normative reference values, which is a table with means, medians, and percentiles for all these uterine features, and you can see them all on the figure. So this data is the new evidence-driven classification. But now there is a problem with this manual approach that the nurse study used. Now it is, of course, it was critical and very important, but it has significant limitations. And even though the clinicians do start with the 3D ultrasound volume, then the actual measurements are taken on specific 2D planes. And this process is extremely time-consuming and labor-intensive, and it is inherently subject to human error and inter-observer variability. So different experts might measure slightly differently. And more importantly, this approach fundamentally simplifies a complex 3D object into a limited set of 2D measurements on a single plane, which means that it doesn't really capture the full nuanced 3D shape of the uterine cavity. For an expert to analyze the complete 3D shape for every patient, of course, is impossible because it is far too time-consuming and not practical. So if we want to truly understand 3D shape, we needed a fully 3D automated approach. So that was kind of the goal of my part of the project. We wanted to take this process from manual to automatic. So the work builds directly on this foundation of the nurse study, and we provided the tools for them to be able to scale up their analysis. So we wanted to build a system that could do two key things. First, it could automatically segment the uterine cavity from the 3D scan. So segmentation, I know that the second speaker will also talk of segmentation, so maybe just the basic definition is the process of identifying and precisely outlining some sort of object of interest. So in our case, this was the uterine cavity. And then secondly, we wanted to automatically align all these different 3D shapes. So we put all the segmented shapes into one common coordinate system that we can then properly compare and average. So let's start with the first step that is often almost the most important step in any kind of science research, which is the data. So for any machine learning project, you need good data and you need a lot of it. So our first step was creating the uterus dataset. Now these datasets consist of 298 3D ultrasound volumes from women that we put in 3 groups. So there was a general healthy population, women with unexplained infertility, and women with recurrent miscarriages. And then to train our model, we manually annotated 141 of these volumes. And this annotation process created some ground truth that then the model can learn from. And this was a very challenging and time-consuming task, which is kind of also the point of all this modeling, because we want to speed up something that takes a long time. Because the quality of the ultrasound images varies a lot. You can also see from these examples, some have a lot of speckle noise or maybe some artifacts, some low contrast, and it makes defining these uterine borders quite difficult, especially, for example, the cervix and where the cervix starts and where it ends. So that was quite hard work, but so we did not, that's why we didn't manually annotate all 298 of them yet. But we have this 141, and we are trying to expand it slowly. So yeah, the key point that Bianca already mentioned is that we've made this entire annotated data set publicly available on GitHub. And we hope that this will help other researchers develop new tools in this field, because there's a lot of applications that could use this. So once we had the data, we trained an AI model for automatic segmentation. So after evaluating several different deep learning architectures, we chose NNUNet. This is a quite basic, not basic, but standard state-of-the-art model for biomedical imaging. It is a type of a 3D UNet, so convolutional neural network, that is self-configuring, which means that it can automatically adapt its own parameters to get the best performance on a specific data set. Which is very useful for anyone that just needs some sort of a model that will work on their data set. So we trained it on our 141 annotated volumes, and it learned how to identify the uterus in the new unseen scans. We did use quite a bit of data augmentation. So we added artificial noise or changed the brightness of the volumes to make the model more robust and able to handle this wide variation that we see in real-world ultrasound quality. And the performance that we got was quite good. So we achieved a dice coefficient of 0.9, which means that there's a 90% overlap between the model's prediction and the expert's manual annotation. Now, to confirm its robustness, we also tested it on scans from a hospital that wasn't in a training set, and it still performed quite well. It was worse than if we used it on a volume that was from the same hospital, because everybody has different machines, or they have different settings on the machines, or there's even if there's a different technicians taking the ultrasound, they can look quite different, but it still performed quite well. So here on the right side, you can see an example of a good result. So in the figure on the top, the blue is the ground truth, and the pink prediction, that is the prediction, are almost identical. And then on the bottom, you can see the predicted shape and all three axes in the 3D volume. Maybe you cannot see it very, very well, but yes, kind of. So after the segmentation, we have these 3D shapes, but then each one is in a different position and orientation within the scan volume. And to compare them or to create any sort of an average shape, they have to be aligned. We found that the traditional alignment methods like ICP didn't work really well, because the uterine shapes vary so much between individuals that we cannot just say, yeah, we cannot use any of these simple methods. So we developed a new multi-step method that uses geometric principles. So first we make a rough alignment that's using just the object's bounding boxes. Then we computationally flatten this natural curve of the uterus. And we do this because this curvature itself doesn't have any diagnostic significance, but removing it allows for a much more accurate comparison of the core shape of the uterus, which is what we are actually interested in. And then finally, we use the principal axis of the shape for some fine-tuning and trying to really align the shapes as much as possible. Now here on the right side, you can see a comparison between a manual alignment of 10 shapes on the top. These were done by the gynecologists that we are collaborating with. And then on the bottom is the result of our automatic algorithm. And we can see that while there are some differences, the results are still quite similar, and the automatic alignment still does align the shapes quite well. So we can produce some clinically relevant orientations, but with a lot less work. So after we run our full pipeline, so from automatic segmentation and then the automatic alignment on all the 3D scans from the general population group for now, we can generate the final result, which is the average 3D model of the uterine cavity. So here on the right, you can see that, which this is just overlapped and average values. And the shapes, and this is completely automatically derived from the dataset. and each individual shape was identified and oriented just by the algorithms and then combined to create this model. And we can see that the resulting shape does have this clear delta configuration, which is consistent with the clinical observations of a healthy uterus. And this result means that our pipeline is able to just input a raw 3D scan or a multiple raw 3D scans of uterus and then produce this clinically relevant average 3D shape. And this does capture more detail than a model that is based just on those 2D measurements. So to summarize a bit, we have built this automated pipeline for analyzing 3D uterine ultrasound data. Now, the main benefits are that it saves a lot of time compared to the manual approach, which is usually why we need the AI. It is consistent and it does remove human variability. And all this means that it makes large-scale research possible. So now we can realistically analyze thousands of shapes, not just hundreds that we were able to before. So this tool can be the foundation for the next phase of research, whatever that may be, because we do always need data in science to do any kind of research. So we can use this tool or just parts of this tool for anything that we want. Now for the future work, the first step is definitely to move beyond just making these simple averages that we are using for visualizations now and use these resulting 3D shapes to build some more detailed statistical shape models. And this will then allow us to compare the shapes of different patient groups. So we want to compare the healthy population versus those with unexplained infertility or recurrent miscarriages. And then the ultimate goal would be to see whether our 3D analysis can uncover some new subtle shape differences that are associated with these conditions, and potentially lead us to some new clinical insights that maybe traditional measurements or traditional ultrasounds might have missed. So this would be it from me for now. I just made a short presentation. And of course, I'd like to thank my co-authors and all the collaborators from the NURSE study for their work and especially for providing all the data that we have. So as a reminder, the uterus dataset and the trained models are open source. They're available on GitHub, so you can check the QR code. And the original paper of the NURSE study has recently been published in Fertility and Sterility, so you can also check that one out. And this one on the top is the paper that we published with the data. So yeah, this would be it for me. And if anyone has any questions, I am open to it.
Thank you so much. That was super, super clear and very on time, which is rare. So thank you very much, Eva. I let Christine take over and present our second speaker, Roland, and we'll take the questions at the end. Thank you.
Thank you.
Thanks, Bianca, and thanks, Eva, for the first talk. So our second talk will be given by Professor Roland Brock. Roland Brock studied biochemistry at the University of Tübingen and Chapel Hill, North Carolina, in the US. And following his PhD at the Max Planck Institute for Biophysical Chemistry in Göttingen in 2002, he became the head of an independent research group in Tübingen. And in 2006 was appointed as a full professor in biochemistry at the Radboud University Medical Center in Nijmegen in the Netherlands. His research aims at developing nanomedicines for targeted therapy. In this context, he uses advanced microscopy techniques to understand cellular processes at a molecular level. And being a researcher in a clinical context, projects are directed at pathologies in need for more effective therapies. And I'll turn it over to you, Roland.
Well, thanks a lot for this kind introduction, and also thanks a lot for the invitation to present here. Yeah, you may ask yourself, why is a biochemist, which on top of that is working on nanomedicines, now presenting a project in artificial intelligence-based tissue segmentation and cell identification in multiplex stained histological endometriosis sections? Well, it was already mentioned that we really do our work in clinically relevant contexts, and endometriosis is a highly interesting relevant context. And if we want to have an effect in a complex environment or in a complex disease, we certainly also need to be able to assess an impact. And that has brought me then to this AI-based image analysis project. So let's see. Yeah. So I don't think that in a gynecological context, I have to give an extensive introduction into endometriosis. You know that endometriosis is the growth of uterine tissue outside the uterus, and this then happens primarily in the peritoneal cavity. But there are different isoforms or different subtypes of endometriosis that can be distinguished based on the site of location. So we can distinguish superficial lesions from deep lesions, which are by definition deeper than 5 millimeters into the tissue and frequently invade muscle. And then the endometrioma or ovarian cysts that, as the name says, form cysts inside the ovaries, which can also have grave effects on fertility. And then we can address this at a cellular level. And now, by going from histology to the cellular level, we get closer and closer to my field of action, namely influencing molecular processes. And then it can be appreciated. I mean, we see that we have an epithelium here formed in these lesions, in the pelvic peritoneal endometriosis as much as in the endometrioma. But what is more important is that part of these lesions is also a quite complex immunological ecosystem. Now, these lesions clearly show differences in histology, and here you see some literature studies that we compiled or to illustrate these differences based on the contribution of stromal cells and epithelial cells. What you see down here is unpublished data that we did on infiltration of immune cells, where we also noted on a qualitative scale at that point some differences in immune cell subtypes. And so lesions are really a cellular ecosystem where, and that is also part of the whole lesion process, where namely this foreign tissue that implants and that is subject to the hormonal cycle, because of this hormonal cycle and the induction of vascularization and so forth, induces a state of inflammation. And this inflammation is associated with the infiltration of various types of immune cells that are also causative then for the tissue changes as a consequence of the inflammation. So we have changes in natural killer cell populations, neutrophils, T lymphocytes, B lymphocytes, dendritic cells, but very importantly also macrophages. And it is these macrophages that play then a critical role in the induction of fibrosis. So that underlying to this epithelial layer, we have changes happening in the stromal cells, such as epithelial to mesenchymal transition. We have fibroblast to myofibroblast transdifferentiation. So we have accumulation of fibrotic tissue, scar-like tissue, which then is also associated with the symptoms of endometriosis, such as contacts of tissues with each other that can lead to pain, or also the inflammation in ways that are less understood, influencing fertility. So when we look at such a lesion, the questions that we ask ourselves are, can we quantitatively capture lesion-specific differences? What is then the lesion-specific cellular ecosystem, and what are the functional relationships of cells? How do these characteristics change with the aging of lesions? Assuming that we go from an early stage of early engraftment to a later stage where we have more accumulation of fibrotic tissue. And then very importantly, and this is then where now I'm concretely involved with research projects, what is the impact of therapeutic interventions? And so that brought us then to our AI project, where we followed a two-level approach, both involving segmentation, as just mentioned, by Eva. So one is what is the overall tissue composition, and the other one is which cell types are present and in which spatial arrangement. And this work we have then, it was published at the beginning of this year in Human Reproduction. So in what I'm going to show you is more or less a pilot study, but I think where we nicely implemented some fundamental principles that will also be guiding for future research. Namely, sorry, that's my coffee machine that is switching off. The idea that can we first of all distinguish key tissue markers? And so we looked at a number of markers: calretinin for mesothelium, so the cell layer, CD10, and vimentin for endometriosis stroma, alpha-SMA for myofibroblasts and smooth muscle cells as indicators of fibrosis, and then CK7 for endometriosis epithelium. And then we have two markers that I highlighted in red, which are not so much discriminative of tissue substructures, but which we included as functional markers. So we had CD45 as a pan-immune cell marker, which we used to demonstrate that within these tissue contexts, we can also identify and quantitate cells specifically, and the same for Ki67, which is a proliferation marker, so that we could ask questions such as how proliferative are immune cells in the context of a specific tissue structure, aiming at determining potential relevant differences. And so the methodology that we used is tissue and cell analysis of multiplex immunohistochemistry. And I thought that I first go a little bit into the basics of immunohistochemistry and multiplex immunohistochemistry. So as you may know, in histochemistry, we use antibodies that detect specific cell surface markers. And then as you do in histology, we use a secondary antibody, in this case labeled to an enzyme, horseradish peroxidase. And now we make use of an enzymatic reaction that, namely a fluorescent dye with a further chemical moiety, undergoes a chemical reaction when horseradish peroxidase creates reactive oxygen species in the presence of hydrogen peroxide. And this activated molecule now reacts with molecules in the vicinity of our primary antibody, and with this we can generate very strong stainings. And the way how this multiplex immunohistochemistry works is that you do a first labeling reaction of your antibody with the secondary antibody. Then you deposit your fluorophore. Then you treat your sample with heat, and this heat will remove the primary antibody and the secondary antibody. Then you come in with another primary antibody directed against another cell surface structure or another cell type. Again, the same secondary antibody, and you will use a different fluorophore so that with the second cycle, you label your tissue with a different color than with the first cycle. And this process you can actually repeat up to 8 times, so that you can deposit 8 different colors specific to specific cell surface structures in your tissue. And so in such a protocol, you first ask yourself with standard immunohistochemistry using standard labels, what is it that we want to see? So you check the suitability of your markers, you optimize antibody concentrations, then you optimize your immune fluorescence, and then you actually establish the multiplex procedure to finally get you to some artistic multicolor images, as you already see here on the right-hand side. And so this is the total protocol. There is a machine to do this, so you don't have to do it all manually, where you have your histology slide, you deparaffinize, and then you go for the first antibody through this procedure. Then you have the heat antibody removal. You do it the second time, again removal, third time, fourth time, fifth time, and in our case, 6th time. And at the end, you can stain all your cell nuclei with a DAPI counter stain. What is specific to this procedure, so it's not that you could do it right away. There is some, with each technique that is simple in the basics, it needs refinement to get to the optimal result. And for example, here, one of the refinement steps that are needed are to test at which place you ideally place your specific antibody. The point is, as I mentioned, with each cycle, your sample is exposed to heat, so above 60 degrees to remove the antibody. And this heat will also denature your antigens. And so antigens that are more sensitive to heat, you should label early in this whole process, because after many cycles, they will be denatured and not recognized by an antibody anymore. Whereas epitopes or structures that are more robust to heat, you can put down in the line, because then also after many cycles, the antibody will still bind them and they will still be recognized. At the end, you have your procedure ready. And so in our case, it looked like that. We started with calretinin, then CD10, CD45, Ki67, alpha-SMA, and in the end, this replaced cytokeratin. So we used a pan-cytokeratin marker, and then the counter stain, and these are the fluorescent labels that we used. And that then gives an image like that. So where you can see, okay, where do we have CD10? Where do we have immune cells? And here you can already tell that the immune cell invasion really differs per tissue substructure and so forth. And so this is then this multiplex panel per channel, where you nicely see, okay, here I have a fibrotic structure. Here I have immune cells scattered throughout. Here I have dividing cells. And then we, in our pilot study, we looked at a total of 9 samples. We had 4 times deep infiltrating, 4 times ovarioma, and 1 superficial peritoneal lesion. And from these lesions, we first took those parts of the image that we could attribute to lesion and not surrounding tissue. And then here you see examples of these multiplex images of different lesion types, where again, you see the different tissue substructures. And so the first step then was the tissue segmentation. And so here, as I mentioned 2 slides earlier, we took out those parts of our images that we could attribute to lesion. And then we had a concept of a lesion where we say, okay, we have fibrotic tissue, we have stromal tissue, we have epithelium, and we have lumen as the most fundamental structures of which a lesion comprises. And then in the first step, we used software that was part of the imaging system called Inform, which you basically manually educate to recognize tissue regions. So you manually draw in parts of the tissue that are epithelium or stroma. And then based on that, you teach your system to recognize and segment these image parts. So in a similar way as we saw in the previous presentation, but now not only for a luminal structure and the surrounding tissue, but for tissue that surrounds the lumen and then the stroma around. So that was the first step. And after having this done, we could generate output in the sense of, okay, if I have a histological section, what is the percentage of each tissue type, epithelial, stromal, fibrotic, mesothelium that is part of the histological section that we see. And then the second step was the cell identification. And for the cell identification, we made use of a machine learning based approach that was established primarily by Mark Gorres at our institute in the context of analyzing cell-dense regions in cancer. And the idea here is that in particular in cell-dense regions, classical image segmentation approaches have big differences of telling cells apart. And so the way how this works is that basically, as we could see, so that is a very nice similarity to the previous presentation, you create your ground truth by manually annotating cells in such an image. So telling, okay, these are CD20 positive cells, these are CD3 positive cells, and so forth, and then really clicking manually. And in this way, teaching your neural architecture where your cells are. And so I don't want to go into details because this is definitely not my area of expertise. There was a neural network with a specific multi-layered structure, and this was then manually educated to be able to recognize cells and then distinguish cells based on these different markers. In particular, also resolve difficult situations as you have here, where you have 5 cells very close to each other, which is a challenge in classical image segmentation approaches, which typically orient themselves in bright membranes. But you see that here there is no continuous round circle around the cell. And so this we also did, and then we compared in our case also this ImmuNet approach to the InForm approach. So the InForm that we used for the tissue discrimination to look at the different cell types also has a cell identification, but we found this is now a little bit difficult to see these red spots here, that InForm had a problem in hyper and hyposegmentation. So there were, in some instances, too many cells recognized, that is hypersegmentation, and in other cases, too few cells recognized, that would be hyposegmentation. Whereas, as you can tell here, the ImmuNet very nicely hit the spot and got all the cells correctly. And so I don't want to go very deep now into the quantitative data because that really had pilot character to demonstrate the feasibility of the approach. But in the end, for these 9 lesions that we had, we could then really in an automated fashion determine, okay, what is the percentage of pan-CK positive epithelium, the percentage of CD10 positive stroma, the percentage of fibrotic tissue, and how does this differ for these different lesions, creating now a basis to automatically segment and quantitate tissue types in histological sections. And more importantly, by combining this with the ImmuNet-based cell quantification, we could now also tell cell numbers. And this is now positive. These are now the immune cells for which we had the CD45 as a marker. And now we could really say, okay, these are how many immune cells we have. Which percentage are the immune cells in relation to the total tissue, in relation to the epithelium, in relation to the stroma, and in relation to fibrotic tissue. And this now can directly take you into clinically relevant questions, because now you can ask questions such as, is a certain immune cell type related to the induction of fibrosis? Or does an intervention that we apply lead to a reduction of the cell type in a specific type of tissue? So if I conclude here, with the combination of InForm and ImmuNet, we can really identify identify different structures and identify cell densities in relation to tissue structures. We have now taken first steps to really use myeloid and lymphocyte panels to really look in detail into the immune ecosystems that we have. And then the next step certainly will be to analyze the cellular ecosystem in relation to lesion type, fibrosis, and therapeutic intervention. And with that, it leaves me to mention the people that were involved. So from our Department of Medical Biosciences, Mark Gorris as the one with the expertise on the cell identification, Scott Corman as the master student who did the major part of the study, Wouter Verdurmen with expertise in fibrosis. Then from our Department of Obstetrics and Gynecology, Annemie Knapp as head of the department, and Guus Vissers, who contributed expertise on the tissue sample, the Department of Pathology, and then that was also initiated as a collaboration with the Arabian Gulf University. And with this, I thank you for your attention. And now both of us are happy to take your questions.
Thank you. Thanks a lot, Roland. That was fantastic, and I really like those artistic images. Yeah, I don't think AI can generate anything like that yet, so that was impressive. Thanks a lot. I would ask Eva if you can turn your camera on again yet, and if you don't mind, I would probably start with a question that someone put in the chat for you, and while I read that question, Everyone else, feel free to either put your chat in the question, or you're more than welcome to put your hand up and then turn your microphone and camera on and ask questions to our speakers. So the first question that Sylvia actually asked was, how will this model of normal uterus be used in the clinic? Will there be a distribution of what counts as normal versus not?
So I think that coming to the clinic will still take a while, because coming from the research to the clinic might take a while. But the idea would be to see whether maybe something that we have considered normal before is actually not, and it would require some sort of treatment. Because, for example, for the septate uterus, I know that the classifications for that have been different. So somebody measures it differently than someone else. And then the ones that still think that's a normal uterus do not perform any treatment on it that they maybe should. It's just one of the options. So I think that is how it would be considered in clinic, not just, okay, this is normal, nothing wrong with it, we're not going to do anything, but just to see whether maybe, maybe just to extend what, where we do consider some sort of other treatment or not. And I don't really understand the second part of the question. If there will be a distribution of what counts as normal versus not. I think there has to be another research done down the line, just to maybe do some classifications and see the actual distribution of maybe,
If Sylvia is still around, and Sylvia, if you want to ask a follow-up question or just clarify your question, just more than welcome to do so. While I can't see any, there is another question for you, Eva. If you don't mind, I'll just jump to Roland for a quick one before asking you another question. Roland, I was just wondering about the markers, because at the moment you have a library with a few markers all together, and then you'll get that image with multiple channels. So I assume each color is a different marker. How do you, what do you do after that? What will be the next step in terms of separation of channel, and how that will then be used for the AI?
So the next steps that we would like to do is really use this for the myeloid and lymphocyte panels. And we have done a try so that we can really say, okay, if we look at fibrotic tissue, how many M1, M2, so pro-fibrotic, fibrosis resolving macrophages do we find in which type of tissue? Or if you assume that we do an anti-inflammatory, that patients undergo a treatment, can we see this back in the composition of immune cells in the lesions that we have? This certainly involves taking biopsies, so this is what we depend on. But that would be the next step, that we get a better understanding of what are the functional interrelationships of the cells, and can we, based on these, derive informations on how cells communicate in this context.
In terms of communication, would you be looking at how they would exchange some nutrients, or how they will?
No, the first thing would be a communication based also on neighborhood. So how close are different cell types to each other? And also then we had this proliferation marker, Ki67. So to which degree are cells proliferating in certain tissues? Can we learn something about that? So really a better function in the same way. So here we really think in analogy to research done in cancer and the cancer microenvironment, where people now ask questions at to which degree is the cancer microenvironment immunosuppressive? What does that make with the immune composition? For example, infiltration of regulatory T cells over other T cells. How is this influenced? So very much an analogy to these type of analyses, approach lesions in much more functional detail than is typically done with the histology that is done for the major part right now, where clinical histology mostly addresses the point, is there suspicion of malignancy, yes or no? And this is what you want to learn. And in that way, I think, or we think that you can gain a lot more on endometriosis biology if you better look at the cellular ecosystem.
Totally agree with you, especially with it will just be probably a kind of easy translation from the existing models applied to cancer to these images. But yeah, that's promising then. Thank you, Roland. Eva, there is another question for you from Lorraine. Lorraine, would you like to unmute yourself and maybe ask the question yourself?
I can also just read it and answer.
Oh no, if not, yeah, just usually give people 1 minute, just because usually they go, okay, no, it's fine, I'll do it then. So the question is, what else is known about women with these conditions? I wonder if the uterus will tell us other things. Another question is, love to explore more about the different outcomes when used from different hospital data sets, etc. It actually will be interesting to know, how do you see this translate to different hospitals with the different data sets, if you could comment.
So maybe first thing, we know the general things about these women. So we have the ages, we have for the groups with the multiple miscarriages, we have the number of miscarriages and things like this, but we did not include any of this information for now. We could, of course, do that later on if we want to figure out something else from it. One major downfall or like downside of this data set is that it doesn't have a lot of diversity, because it is this general population that we have is the general European population still, right? So it would be great if we could expand that to other races, other ethnicities and everything else. And the other question with the different hospitals data set, so we found the biggest difference was actually in the type of the machine itself, or in the brand of the machine. So we mostly used GEs because most of the hospitals that were included in the project had GEs. So when we tried translated it, for example, to a Samsung machine, it didn't really work well. But just using another hospital that still had the GE machine with the same kind of, at least the same age or like the same resolutions and stuff, it was quite okay. So just the difference between different operators wasn't as big as we thought it was going to be. But we do want to include more of the Samsung machines as well, because I think GE and Samsung are the most popular. So that people with Samsung machines can also then use this model to predict their own.
Yeah, that's interesting. I think you touched the very important points, which is the bias of the AI models, which of course, if they are trained on specific data set, they will never be able to predict something that they've never seen. Which is a recurring theme in our conversations. I also had another question for actually both of you. And I was wondering if you could comment a bit on how do you see the field moving forward in terms of AI applications in your specific field. So if you'll say, what will be the next step or the next challenge for the AI community to take on board to move forward your field, what would you say that will be? I don't want to put you on the spot, so just whoever feels ready to start.
Well, I think there are different levels. So if we look into histology, I think then the really analysis of complex cellular interplays, that will be more and more important. And that certainly goes along with techniques also such as single cell sequencing and so forth, that people really aim at understanding the enormous diversity of cells and also the continuity of cell types, where these very specific subtypes that we now use as classification schemes may increasingly disappear or at least become gray boundaries. Then if I comment on a field that is absolutely not my expertise, but where I am getting bits and pieces, then it's certainly also that for endometriosis diagnosis, Also, laparoscopy is now increasingly replaced by advanced imaging techniques. And I could imagine that there, what we just saw from Eva, or also feature recognition, AI-supported feature recognition in MRI or ultrasound images could also gain grounds.
Yeah, I think here it's very important to just have as much data as possible be open source so everybody can gain from it, because AI, of course, needs data. If we don't have data, there's nothing there. So if we do get a lot of ultrasound data, then I think a lot of AI imaging analysis have still a lot of potential to help with either diagnosis or maybe even treatment in the future. So yeah, I think there's still a way to go.
I think in your slide, Eva, you mentioned about the challenges though about ultrasound, that they might be totally different because of different centers use different equipments or just by the way the images are saved. How challenging you think would be then to find or to develop a single model that could actually work with those modalities?
I mean, here in our use case, if you have like one study, you usually have some sort of standardization. So you tell the people how they need to take the ultrasound, and then you have some sort of like an idea what they will look like. So yeah, maybe in a real world scenario that wouldn't really work that well. But I think in in AI in general, if you have a lot of data, you can also overcome that. But we are not yet at a point where we have enough data, especially open source.
Yeah, that's actually true. Yeah, and thank you for sharing that link. I'm sure it will be very helpful for the community. Going back to the point that Roland mentioned earlier about the images where you can get some information about the interplay between cells. Now there is the spatial transcriptomics techniques that, of course, in cancer is going much faster, I would say, than endometriosis. But there's been a recent work that actually is published as public available spatial transcriptomics for endometriosis. And that will mean that there's going to be the possibility of integrating multiple modalities for understanding interplay of cells or even for diagnosis. How do you see that moving in terms of modalities? If you had to pick which modality would be more important for endometriosis or for diagnosis, for example, which one would you?
Yeah, I would not dare to make a prediction, really. I think the main problem with all these analyses for me is now which will in the end give us actionable data. So I have to say, when I see these single cell transcriptomics analyses, which then translate into UMAPs and so this highly complex data, I see a lot of description, and I ask myself then about the extra level of understanding. And I think that will be the big challenge for the future, that we go from description. So now we may be at the point that we understand that there is a continuity of different cell phenotypes which may have functional relevance. That is probably the understanding right now, but then translating it into a functionally relevant understanding, which can then also lead to actionable insights in terms of what we will now really do that changes that in a meaningful way, or how can we use this highly complex data in order to make testable hypotheses. This is where I see the main challenge. And for me, this is where then also the proof of the pudding lies for the different techniques. Which of the techniques will in the end be used in translational research to feed hypotheses and get to novel treatments or better diagnosis. And this I wouldn't dare to predict now.
Do you want to add anything on that, Eva? No, it's fine, yeah. Because actually that takes to my next and final point about, you mentioned UMAP, which is what we tend to use at the beginning to probably try to make visible, like make 2D the problem that it's actually multiple and high dimensional, so just It's a kind of comfort type of plot that we tend to use just to say, okay, so we can visualize the data, then what can we do with that? But that's actually raises another important point, that we have the AI community on one side that keeps building models, and on the other side, we have the clinical and biology and bioscience community. And as you were mentioning before, I think that's the only way to move forward, is just to make sure these two communities are collaborating each other, because it's not going to be possible just to do everything one single lab in one single center. I think the key for moving forward the field is really collaborating, which both of you were collaborating with the other either clinician or AI expert, which both of your works are very good examples of. I just want to finish with that, and I'd like to really thank both of you, Roland and Eva, for your insights today. This meeting has been recorded, and Emma will just do a bit of cleaning, and then we'll publish on our YouTube channel. We do have another meeting around December time. We're just finalizing the dates, and we will share this soon with the community. And I'd really like to thank you again one more time, and have a nice rest of the day, everyone. Thank you.